Yeah, hi everybody. My name's Dr. Harry Leider. I'm the CEO of Organicell, and we're delighted to have you tonight to spend a little bit of time talking about just perhaps one of the most exciting areas in medicine, and certainly one of the most exciting areas in regenerative medicine. We're going to talk about exosomes. What we'd like to do is have you um, uh, listen to the presentation, but if you have questions, and we hope you do, pop them into the chat. There's a chat button at the bottom of your screen, and we'll handle the questions at the end of the presentation if that works. But I also want to pitch this to Dr. George Shapiro, who's one of the foremost experts in regenerative medicine, and maybe he can make a couple of comments before we get started.
Sure. Thanks, Dr. Leider. So I wanted to just welcome everyone. Some of you may know me from the AMMG. I'm on the speaker panel. I've been with the AMMG, Age Management Medicine Group, since 2006, and been going to all the conferences every year. It's probably one of the best age management conferences around. There's two main organizations, but this is probably the premier one. I want to thank Greg Fillmore and Rick for setting this up. It's going to be about 40 minutes of education on exosomes and some of the new things that are involved with exosomes, and then we're going to do some question and answers. So, Dr. Leider, you want to start?
Sure. Thank you, George. Sorry for the slides self-moving on their own. Probably AI was doing it. Anyway, this is the group from Organicell that's going to be speaking to you tonight. George introduced himself. By way of background, I'm a general internist by training, so I'm not trained as a regenerative medicine doctor, but learned a lot as the CEO over the last 7 or 8 months here at Organicell, and came to Organicell because I felt this was clearly one of the most exciting areas in all of medicine. I have a background running different types of healthcare companies. Maybe Dr. Golub and Dr. Bellio can introduce themselves as well.
Sure, I'll go ahead. Nice to meet you all. My name is Michael Bellio, and I'm the laboratory director at Organicell. I manage the research and manufacturing for the team. And just quickly, my background, I have a PhD in cellular pharmacology, and a lot of my training and research has been focused on cell manufacturing for different types of cell-derived products for clinical trials and other types of clinical applications. So happy to be here to help with the discussion.
I'm Howie Golub. I'm the Chief Science Officer of Organicell. I have about 40 years background in all kinds of things, but specifically clinical research development. And this is an extremely exciting area for which to do clinical research in. So I'm really excited to be part of the company.
Great.
And I'm George Shapiro. I'm a cardiologist. I trained in New York with a focus on heart transplant and stem cells. Did a lot of research on it, joined Organicell and became their chief medical officer about 7 years ago. And we're excited about some of the new research we're going to talk to you about tonight.
Great. So thank you. So here's what we're going to do tonight, and we're going to try to move through it pretty quickly so we can get to the Q&A and hear what your thoughts are. We're going to talk about the history of exosomes. Some of you may know something about exosomes, others may know less, but we're going to go back and cover it so everybody's on the same page. We're going to talk about the science and mechanism of action of exosomes. We've learned a lot over the last 7 or 8 years in particular. We're going to cover the regulatory landscape of exosomes. This is a big topic for those of you that are thinking about using exosomes or using exosomes and how the FDA thinks about them. We're going to talk about the clinical applications, and Dr. Shapiro in particular has a wealth of experience to share here. And we're going to talk about the next generation of exosomes, which is really the idea of autologous exosomes. Exosomes derived from a patient's own blood, isolated, and then used with the same patient, which is the next generation cutting edge. We'll go into that in a lot more detail. So just for a little bit refresher on exosomes, extracellular vesicles are these cell-derived membranes. And you look on the right, that's a cell, of course. And these cell-derived membranes are the blue bubbles, and the Golgi apparatus and the different mechanisms within the cell secrete into these vesicles all sorts of bioactive proteins. Important is probably microRNA, but there's also DNA and RNA and lipids and other cell proteins. And the microRNA actually influences the activation of RNA. And of course, the RNA affects the DNA transcription. So microRNA is very important. But these EVs, many of them are actually exosomes. And these exosomes are present in nearly all body fluids. They get secreted into the blood. They are a way that cells communicate with each other. And what's really interesting is up until about 7 or 8 years ago, we thought that exosomes were kind of like the garbage system that got rid of debris and toxins out of the cell. Now we know that all these bioactive compounds like microRNA actually have a potent effect on reducing inflammation and causing some cells to regrow, things like cartilage or hair or skin. So they're very small. They're 4,160 nanometers, and a way to get your mind around that, because I can never really think about nanometers, is they're about a thousandth the width of a human hair. That's how small they are. And they cross membrane barriers, including the blood-brain barrier, which is really interesting because, as you know, most drugs don't cross the blood-brain barrier. And exosomes really are the next generation of healing because they're the body's own natural mechanism to damp down inflammation and promote cell regrowth, whether it's in the skin or the hair or the lungs or the kidneys. So it really is an exciting area because the downsides of exosomes are extremely minimal in terms of side effects or complications. So, there's been just an explosion of research. I'm not going to go over all these articles. I'm going to go over some of the categories of research that are just booming right now. Lots of mechanism of action studies talking about exosomes reducing inflammation and causing cellular regrowth. Lots of research in the areas of cardiology, repairing damaged heart where there's scars and there's heart failure, pulmonary complications, especially due to viruses like COVID. Neurodegenerative, this is being looked at for things like Parkinson's and Alzheimer's. And actually, we're quite interested in some of the neurologic complications as well in our company. Anti-aging, this is being used with just the general thinking that if you give someone exosomes, it may actually reduce the effects of aging across the body, and especially in the lung. And then there's a lot of interest in the orthopedic community, the sports medicine community, the rehabilitation community around bone diseases. Probably the most common, and we're going to talk about a little today, is knee arthritis. But the whole idea of getting the chondrocytes to regenerate when there's arthritis is a big area of research, and we have a lot of practical experience at it in the real world, and George is going to talk about that a little later. And then what's the future? Well, this whole idea of personalized medicine applies to exosomes too. There's a lot of research in looking at exosomes as biomarkers of disease. And maybe you can look at exosomes in someone's urine or blood and pick up a disease before you would pick it up in other ways. And that there may be ways to use different types of exosomes for different patients' conditions. Maybe we can use AI, and we're working on this a little bit, but there other companies are too, about AI to tailor exosomes and load them with specific cargo, perhaps medications or drugs. So this future is really exciting. So we now call this the secretome, which includes exosomes and other microvesicles. And again, the big idea here is reducing inflammation, repairing damaged tissue, and delivering this messenger microRNAs to the body. And I want to talk a little bit about inflammation, which is one of the two big ways that exosomes work: inflammation and cellular growth and repair. And you may know that monocytes get converted to macrophages. And there's two types of macrophages, basically. There's the M1 macrophages, which are pro-inflammatory. They're bad. There are we call polarized. They get converted when there's inflammation to M2 macrophages, which are anti-inflammatory. I'm sorry, not when there's inflammation, but they can be converted to M2 macrophages, which are anti-inflammatory. So M1 are inflammatory, pro-inflammatory, M2 are anti-inflammatory. And the key thing to remember here, the punchline is exosomes can convert M1 macrophages into M2 macrophages from pro-inflammatory to anti-inflammatory, and they can work throughout the entire area of the body as you see below. It's all the different organ systems. Now, let's talk about what are in exosomes. Well, there's 300 bioactive chemicals, but there's growth factors, cytokines, messenger RNA, microRNA, and these are expressed, many of them on the surface of the exosome, some internally and some on the surface. And one of the ways we can identify exosomes in the body is these surface markers like CD9 or CD63. Why am I telling you this? That later in the discussion, we're going to talk a little bit about how we can measure the number of exosomes because of these surface markers like CD9 and CD63. But in general, the role of these exosomes are to basically modulate immunization lower, cause cellular regrowth, and to have anti-apoptosis effects. Now, the therapeutic effects of exosomes, as we mentioned, can really be almost every organ system of the body. And I'm not going to go into all the different mechanisms of action, but it can be in the heart, it can be in the liver, it can be in the kidney, it can be in the brain, it can be in the skin. But these basic ideas of damping down inflammation and cellular regrowth apply in almost every organ system of the body. So what I want to shift gears for a moment, sorry for the slides migrating, to this idea of autologous exosomes. So there's two ways to get exosomes. You can get them from one person, from a donor, through amniotic fluid or placental tissue or Wharton's jelly or mesenchymal stem cells or adipose stem cells, and then isolate exosomes and then give them to the patient. That's allogenic, of course, when you take it from one patient and you give it to another patient. But this breakthrough that we're very excited about is the idea of autologous exosomes, taking a subject or a patient who's got arthritis, or you're treating for hair regrowth, or you're using it for aesthetic purpose or maybe sexual medicine, getting their plasma and using a proprietary process, isolating billions of their exosomes out of their blood, kind of like PRP, but not platelets, exosomes, and then giving the exosomes back to the patient wherever they're needed, in the joint, in the skin, in the hair follicles, wherever it's needed. And as we talk about autologous exosomes and why they're important and why we think they're a next generation regenerative medicine, they overcome some of the limitations of platelet-rich plasma, which of course is very popular. Many of you may use platelet-rich plasma and certainly know about it. We're going to talk about how the mechanism of action of autologous exosomes, that's what AEs are, is potentially superior to platelet-rich plasma. We'll get into these regulatory considerations of why are autologous exosomes potentially advantageous to you in practice from a FDA compliance standpoint, and then we'll cover some of the clinical uses of these autologous exosomes. So I really cover this. You take plasma, number one, from a patient, and we isolate out in a multi-step proprietary complex process that includes a high-speed centrifuge that runs at 20,000 gravities. That's the force of the centrifuge. It's not a PRP centrifuge you have in your office. It's a big machine. It costs a lot of money, and it generates 20,000 gravities of force when a rocket ship goes up and an astronaut is facing the maximum force that before they might black out, it might be 10 or 12 gravities. This is 20,000 gravities. But because of that force and other things we do, we can isolate hundreds of billions of these extracellular vesicles per milliliter. Because of this process, there's no cells, there's no platelets, there's no cellular debris. And from a regulatory standpoint, that's very important because that means it's minimally manipulated, which is one of the reasons we'll discuss in a moment why autologous exosomes do not require FDA approval. It's also one of the reasons why they're not antigenic. PRP can cause a flare, can cause inflammation in some patients initially, and that's because of the antigenicity of PRP inherently. Autologous exosomes doesn't have that antigenicity. In fact, usually the patient feels better very quickly. So we can use or anyone can use a machine called a nanocyte assay. And what this does is it measures the number of exosomes and the size of the exosomes. And on the right, what you can see is these autologous exosomes, at least in our samples, are hundreds of billions of particles. And then their average size is below 100 nanometers. That's on the x-axis. So these are very, very small. They're at the very small end of the spectrum of anything in the body. And this, using bioassays, we can actually measure the microRNA in the exosomes. And you can see there's all sorts of microRNA and other bioactive compounds in the exosomes, hundreds of them. And with respect to one disease, knee arthritis, what this basically is a study that shows that in knee arthritis, if you look at in each graph on the bottom, the controls are on the left, the knee arthritis patients are on the right. They have lower levels of 3 types of microRNA, 26A, 26B, and 27B. You don't have to remember that. The important thing here is that exosomes can replace the deficiency in those microRNAs, and this may be the way exosomes actually reverse the cartilage damage in knee OA. Just a case study of how exosomes can work physiologically. So let's talk about autologous exosomes versus PRP, because everybody knows a bit about PRP. The limitations about PRP is it can be effective. It's used a lot. Some patients really benefit from it, but we know that it doesn't work for everybody. No therapy works for everybody, but because PRP is not standardized, patients are different ages. They have different medical conditions. They may be smokers. They may have diabetes. They may have immune problems. They may be on drugs. They may be taking anti-inflammatories. All these things can affect PRP. And PRP is often antigenic, as we talked about. The other last thing is you really can't store PRP for very long. Usually you got to give it the same day. So that can be a limitation if you want to give patients multiple treatments over time. Now let's talk about knee arthritis again. All of you as clinicians probably remember this from medical school residency or maybe your practice. Healthy knees on the left, you got the bones there, of course, the femur and the tibia. You've got the nice healthy cartilage, articular cartilage and meniscus, and you got a nice joint space. On the right, you get knee OA, and it affects 45% of adults, knee OA, in their lifetime. You get erosion of the cartilage, you get bone spurs, you get narrowing the space, and then a lot of people get this thing called bone on bone, like on the edge here, where the bone's touching the bone. This affects tens of millions of people a year. And, it's interesting. I remember some people saying, well, it's really, the surgery is really not a bad surgery. So, we can just fix knee OA. Well, the reality is that's not true. 15% of people that have a total knee replacement don't go back to work. 20% of people actually have surgery on the other knee within a year, and 20% of people have chronic pain. So it's not a benign condition, even if you do surgery. So the idea of intervening with something like exosomes and avoiding the knee surgery is really a good thing. So this was a recent study that compared PRP to placebo for knee osteoarthritis. It was over a year, and 144 patients got PRP 3 times, and 144 patients were randomized to get placebo. And after a year, they looked at pain scores, and they scanned them, and they looked at the cartilage. And the punchline was, in this one study, the two were not different. Now, certainly within this study, some patients got better, but on average, there wasn't a difference, right? When you do the statistics. I want to acknowledge that other studies are more positive. This was a particular one that's recent and pretty well done. So I wouldn't overstate it and say PRP doesn't work, But I think the punchline here is that it doesn't benefit some patients. And the medical associations have been a little lukewarm about solidly endorsing PRP in some situations. And this was a recent review article that's talking about extracellular vesicles or exosomes being a superior alternative to PRP. So there's a lot of discussion about this being sort of the next generation of PRP. Not that PRP is bad, but this is the next thing, just like a new antibiotic or a new procedure. This has some advantages. And why the mechanisms of action should be superior? Well, we've talked about some of them. There are more growth factors and exosomes in autologous exosome preparations, if they're done correctly, than there is in PRP. PRP has some exosomes. These small size exosomes goes across cellular barriers, cell membranes, into joints, into the brain. It is acellular. It doesn't have immunogenicity. We've covered that. It has more bioactive molecules in it, and there's no cellular material. So these are theoretical reasons that are being discussed as to why it seems that exosomes are the next generation and a bit better than PRP. Now, when we talk about autologous exosomes, we can use that same nanosite technology. And on the left, again, you can see that there's hundreds of billions of particles. And on the right, the way we can actually detect the particles, all those little dots on the exosome are CD9. And if you tag the exosomes with CD9, you can count them. And you can see the bar graph, that's 300 billion. 10 to the 11th is 100 billion. So we can measure that we can isolate 300 billion per dose exosomes using these CD9 assay markers. And then we did a direct comparison, small number of patients, and the research is early, but on the left, what you see is the concentration of exosomes in PPX, that's our autologous exosome, that's just our name, is 2.36 times PRP, and that's highly statistically significant. And on the right, again, using this CD9 fluorescent labeling, we can show that the red dots are concentrations of exosomes, the blue dots are not. You can see that there's a much higher concentration of exosomes in the autologous exosomes, the PPX and PRP. In fact, the percentage of the nanoparticles, these vesicles that are tagged for CD9 are only 14.2% in PRP, and at least in this one patient, they were 3 times as high. They were 42% higher. So again, more exosomes, more tagged with CD9. So we talked about this monocyte macrophage inflammation. I covered it. Monocytes either getting converted to M1 pro-inflammatory macrophages or M2 anti-inflammatory. And in the lab, what you can do is you can take monocytes and you can expose it to lipopolysaccharide. And the lipopolysaccharide activates the monocytes and makes them M1 pro-inflammatory macrophages, and they secrete all sorts of things, TNF-alpha, IL-1B, IL-6, IL-12, all these inflammatory biomarkers. And what we did is we looked at these macrophages that were angry, that were secreting IL-6 and IL-1B. And what you have on the left is a comparison for the IL-6 RNA expression, not the protein, but the RNA being activated that makes IL-6. The control has very little IL-6 inflammatory marker. After the lipopolysaccharide is added, you see the red, and we normalized it to 100. So it had 100%, we just called it 100%, whatever it was, activation. Then with the PRP, you see a slight decrease in the IL-6 expression, but then you see a much more marked decrease in the IL-6 with the autologous exosomes, the PPX. These were 10 patients. On the right, we looked at IL-1B actual secretion, the actual marker, the actual protein, and it's the same pattern. Control has very little of this bad inflammatory marker. 100%, we called it 100% for the lipopolysaccharide by normalizing it. You see a decrease of somewhat with PRP. You see a more significant decrease with autologous exosomes. So that's really some bench research that shows this anti-inflammatory effect of autologous exosomes. Here's another experiment. This is preclinical. This is in mice. And, what happened here is this is a model. This is a preclinical model of mice that are induced to have a osteoarthritic type problem with a toxin. The toxin's monoiodic acetate, if I say it correctly. So let me walk you through it. These are tissue stained of the knee in mice. And the control is the upper left, okay? And the little triangle shows you a nice femur where it's the safran and O stains red for the surface. And you got a nice cartilage, you got a nice joint space. MIA on the upper right is after the toxins put in. You see lots of fibrous blue tissue. You see cellular debris. You see blue inflammatory cells. Then after being treated for 3 weeks on the lower left with human stem cells, you see the inflammations going down. Oh boy, okay. It has a mind of its own. You see the lower left, you see it's looking a lot better. The joint space is coming back, but there's still cellular debris and it's not normal. But look, here's the take home. Look at the lower right. That this is with exosomes, 3 weeks of exosomes, like science fiction. Very, very thick cartilage. Cellular debris is going away, nice thick joint space. It almost looks better than the control. This is just 3 weeks of exosomes. You have the fibrous tissue going away, being covered with the cartilage. So this is another, it's not humans, but it is a powerful experiment. So George actually led this study. This was done at Fountain Life, and it was an IRB-approved study in humans who got either intravenous or intra-articular autologous exosomes. Again, our version is called, what we do is PPX. And what you see on the right is after 3 months, statistically significant, there was a decrease in WOMAC scores. And WOMAC scores, for those of you that aren't, like me, that aren't orthopedists or sports medicine people, they're the standard pain scores. It's a standardized questionnaire for pain and function and stiffness. And there was an improvement in 3 months with autologous exosomes. What was really particularly interesting is the patients that got IV, not intra-articular exosomes, actually had a decrease in their WOMAC scores over the period of 6 months to 9 months. So we believe that the exosomes basically get stuck on or activated by areas in the body of inflammation. And that's how they have an effect even when they're IV. But isn't that fascinating? You wouldn't expect that. But the billions of particles find their way across the cell membrane or the joint membrane and actually had a positive effect on WOMAC scores. Now, this wasn't controlled, but there's no reason to believe that patients' pain scores would get better over a year without some sort of active therapy. Now, I'm going to shift gears entirely as we get close to wrapping up. There is a lot of discussion about safety and regulation of exosomes. And in order for autologous exosomes to be safe, you want it to be autologous, of course. You don't want it to be classified as human cellular tissue or plasma or platelets. You want it to be acellular. As we talked about, you don't want to add things to it. You don't want to be manipulated. You don't want to add any cryoprecipitant. You want to test it to make sure it's not infected with bacteria. And it has to be done in a very, very regulated laboratory environment. These things sound trivial, but this is all the different things that need to occur in a lab to make sure that the autologous exosomes here isolated are safe to put back in the patient. And this is really important thing. The FDA has said under these regulations, it's 21 CFR Part 1271. You'll never remember that. But it says that platelet-rich plasma or autologous biotherapeutics like platelet-rich plasma do not need to be approved by the FDA because it's like a blood transfusion for the patient's own blood. As long as it's not manipulated, as long as you don't add anything, and there's a whole bunch of criteria in this regulation, you don't need FDA approval. And we've gotten multiple lawyers, FDA experts to opine on this. Just like PRP is not regulated by the FDA, does not require approval, neither do autologous exosomes if they're produced in the way we just described. So this is something you can advertise to your patients, you can use confidently and not worry about whether there's any gray zone or whether there's any concerns from the FDA. And again, it must be processed in a lab that's meeting all the quality standards. And again, PRP isn't always the same. So I'm going to just I'll mention that anywhere you're using PRP, you can use autologous exosomes. So anytime you think about PRP, you can think about that. And they can be used in different ways. I talked about IV, we talked about intra-articular, but you can even do inhaled. In aesthetics, it's being used microneedling, local injections with sexual medicine. And I kind of wanted to have George comment because, he's got more experience maybe than anyone in the country using exosomes in patients. And since the studies are evolving, he might be able to share a little bit of, his experience.
George, thank you, Harry. So over the last 7 years, we've been involved, Organicell has been involved in multiple clinical trials, IRB approved trials that looked at patients with acute COVID, subacute COVID, and long COVID. We've seen, and some of the data we didn't show here, I showed at the AMMG, but we've seen a significant reduction in inflammation measured by blood biomarkers. And these exosomes were not autologous exosomes. These were amniotic fluid-derived exosomes. So when the FDA came up in 2019 about all the new guidelines, most medical practices were concerned about not heeding to the FDA's rules of being under an IRB, so they stopped using exosomes. This is the first time since 2019 that you should feel comfortable using exosomes that are your own, autologous exosomes. So the take-home message tonight is autologous exosomes. Now, the amniotic fluid exosomes that we were using before in all these trials showed a significant reduction of inflammation. I mentioned that in the blood, we saw 5 biomarkers all declined, pain, range of motion, all improved by 70%. We actually started using these for other things besides COVID. We looked at arthritis, and we were starting to do trials with the Mayo Clinic and a hospital special surgery in New York with inflammation. So the big question is, are these amniotic fluid-derived exosomes that we were using before equal to autologous exosomes or second-generation PRP? And what Dr. Leider showed you tonight was it's actually better. It has, the product that we use for autologous exosomes, we've now shown that it can inhibit interleukin 6, interleukin 1, and reduce inflammation. And it has a concentrated form of exosomes. So if you think about regular PRP, and then you concentrate that PRP and bring out 200 billion exosomes in 1 ml and inject it, we're seeing good results. And we're excited about it because now, as a public company, we have to be very careful about what we do. And we want to make sure that we're on the right side of the FDA. So this is legal, and this is great. It's the first time that physicians should feel confident about using a product without having a knock on their door from the FDA.
Thank you so much, George. So as we wrap up, one other thing I wanted to point out is an area where there's for exosomes that is accepted and there's less FDA concern, whatever exosomes you use, is dermatology and medical aesthetics, whether it be wound healing and wounds, whether it be hair regrowth, whether it be pigmented issues or anti-aging. And we've all heard about like the vampire facial that's gotten a lot of press because of the stars. But the point is that when you use exosomes, whether they be allogenic or autologous, and we again think autologous has advantages, aesthetics is another safe harbor, if you will. And it's being used a lot. So the summary, and thank you for listening, and then we'll get to the questions. There's about 10 questions in the chat already, is that autologous exosomes, which we're suggesting are the next generation regenerative medicine and have reasons to be superior to PRP, does not require FDA approval, as George said. It's acellular, so there's no antigenicity. It does need to be manufactured in a compliant lab with special equipment. It can't be done with a little centrifuge in the office, like a PRP centrifuge. And even some of the technologies that are being promoted in the office for autologous exosomes, there's a couple, only produce millions of exosomes, not billions. It's only in a lab with very expensive equipment and a proprietary process can you do it. And then we really talked about most of the other stuff on the right, that this is always derived from the patient and given back to the patient and highly concentrated. So the summary is this is really exciting, and we'd love to answer your questions about. There's some references that you can look at later about the mechanisms of action of autologous exosomes. But I want to thank you for listening, and we're happy to take your questions.
So, Dr. Leider, there's a lot of questions in the chat. You want to start off with some of these? I can read them to you.
Yeah, why don't you moderate it, George, and then we can all.
So one question was about cancer. Do exosomes affect cancer?
Yeah, why don't you handle that one?
So I have a lot of experience with patients with stem cells and the secretions of the stem cells, which are exosomes that contain microRNA. Depending on your immune status, the exosomes can work either way. So we don't recommend exosomes in patients with active cancer right now. There's research ongoing, and there's ways that you can do it through trials, but we're not recommending, in active cancer. The future, though, will be exosome therapy to carry different drugs specifically for cancer and target the cancer with exosome therapy. And that's already in progress at multiple hospitals. We don't recommend it, though, in active cancer patients. The next question was advancing age affecting autologous exosomes. So that's a great question. There's been a lot of research, specifically a lab in California that looked at 10 years of research looking at young exosomes versus old exosomes. It turns out that young exosomes are better. That's why amniotic fluid-derived exosomes were great. The older the exosome are, the less potent there is. What's great about the autologous exosome is there's so many concentrated, 200 billion. You're getting a good, a good solid amount of exosome therapy. Exercise takes your exosomes and makes them incredibly powerful, and makes the mitochondria inside those exosomes optimized. So we're looking at new trials of people exercising before we take out the autologous exosomes.
And George, I can add one more thing to that with our experience doing laboratory research. We've looked at the autologous exosome preparations from various age groups. And so far from my research, I found that all products carry a similar inherent anti-inflammatory effect, no matter what their age is. And importantly, all of those top expressed microRNAs that we characterize that hold those anti-inflammatory effects, they're all held consistent between donors. So that's a very important thing when you think of the exosome's ability to mitigate the inflammation, is they have all of the same proteins, all of the same anti-inflammatory microRNAs, and hold similar effects in our bioassay models.
But the truth is that unless you do a large study across ages, we won't really have a definitive answer. However, just in our experience is what Mike explained.
Another question is this applicable to diseases such as multiple sclerosis? You want me to handle that, Dr. Leider?
Yeah. I mean, my take on that is when we get to very specific diseases, whether it be multiple sclerosis or Parkinson's or Alzheimer's, this is cutting edge stuff. The research is starting to, evolve there. But George, you tell me what you think.
Yeah,
My reading of the literature is we don't know yet.
So part of what I do also with, instead of other than Organicells, I work with a company called Fountain Life that looks at advanced technologies to look at these diseases and find out the why. Why do people get MS? Why do people get Parkinson's? And it turns out that a lot of this has to do with inflammation. Mm-hmm. And everything we've been talking about exosomes is it reduces inflammation. So we've had patients in my practice who had Parkinson's disease and came in for osteoarthritis, and we're in our trial, our FDA trial for osteoarthritis of the knee, and it turns out that their Parkinson's symptoms abated. And for those of you who take care of MS or Parkinson's patients, when you start to have muscle issues, it's hard to walk, your gait is abnormal, you start shaking, you start drooling. These symptoms were disappearing. And, and we were treating the arthritis. So the exosomes absolutely can help a lot of these diseases.
Great. Thank you, George. We've got some other questions.
More of these. So are there exosomes in PRP? I think you answered that, but
yeah, I'll cover that again. There are lower concentrations. If you remember that slide, we have 230, the number is almost 2.5, 250% more, 2.5 times more exosomes. So yes, there are exosomes in PRP, but a lower concentration, significantly lower concentration.
And the nature by which the exosomes are formed from the platelets is somewhat variable depending on the stimulation of the platelets.
Yeah.
So, and that probably explains the variability in clinical effect.
Yeah.
So the next question is, how do exosomes compare to mesenchymal stem cells or mesenchymal stem cell exosomes? So I just want everyone to understand that what makes the stem cell work is the brain of the stem cell, aka the exosome. What makes the exosome work is what's inside, microRNA. That's a communicator. The microRNA communicates with an old cell, infected cell, inflamed cell. So we're talking about stem cells, mesenchymal stem cells, whether they're mesenchymal or placental or bone marrow derived or adipose derived. All those stem cells have exosomes, and all those exosomes have microRNA. The question is, if we can concentrate those exosomes and give you a shot of those concentrated exosomes, that's where you're going to get the best approach. We can give you 100 million stem cells, but the exosomes are not concentrated, and stem cells are not legal here in the US. There's a lot of centers that we visited outside the US that do stem cells. Few do exosomes. No one is doing autologous exosomes the way we're doing it, where we were able to use special equipment to concentrate these and give them in one area.
I think we have a question. What's the cost of each session? I expect that means what's the cost of each patient or each treatment? Is that what I think that's what it means? I don't think on the webinar, we want to be too specific, but I think we can be general and suggest this in general, the process of autologous exosomes makes them a little more expensive than PRP. But one advantage is you get 2 doses, 2 1 milliliter doses, which you can use with the patient in 2 different sessions. So that makes it closer to a comparison. So it is a little more expensive, but as you'd expect for something that's done in a lab under highly controlled conditions with special equipment, it's going to be a little more expensive than something that's done in a doctor's office.
Can children donate their exosomes to their parents?
Oh, that's an interesting question. You could, but then they wouldn't be autologous, right? So is it something you could do, isolate exosomes for a child, freeze them, put them on liquid nitrogen?
That's the old way we were doing it, right, Harry?
Yeah, that's the allogenic. So
it
is technically possible, but you wouldn't be dealing with autologous then.
One interesting thing about the autologous exosomes is you can biobank them. You can go to the office, draw your blood, and store your own exosomes and keep them there, just like your biobank stem cells. And every year we lose 10% of our stem cells. We're also losing exosomes. So if you give back your stem cells and exosomes each year, you're slowing down the pace of aging. And we can now measure the pace of aging with specific testing that we do.
Yeah. And that may be the reason why the exosomes are as good when you get older as when you're younger, at least as Michael said in our studies, because it's probably the concentration you're losing. Exosomes probably are, I'm just hypothesizing, exosomes are potent. They don't lose their potency, but you don't have as many of them as you get older, as George said. But by isolating them and centrifuging them, you're giving someone back the billions in a concentrated way.
So, Dr. Leider, I'm going to ask this question to you. This is from one of the audience. I'm an orthopedic surgeon. How do I go about doing this on my patients?
Yeah, yeah. I mean, I can't speak for, I mean, right now we just developed this. This isn't an infomercial, but I can at least tell you that if any doctor's interested in doing this, just contact us after the conference, and we can get you much more detailed information and get you prepared to do this with a patient or two and see, how you feel about the results. So it's really, really simple. Um, do we have a link or some way
If you
people
want, I
follow?
can tell them how we do it at Fountain Life. So,
Sure.
so at Fountain Life, we have 4 clinics. We have PM&R physicians, physical medicine rehab docs who do injections. So what we do is the patient will come in and have their blood drawn, just like PRP. And we take that sample of blood, we send it to the Organicell lab, we check it for bacteria, viruses, etc. We then do a nanosite report to see what's actually in it. Then if you know what, actually, Dr. Bellio, why don't you tell us the process once you get the blood from the patients and it comes to you, what do you do with it?
Well, the only caveat here, Michael, is we don't want to give away all the secret sauce here.
No, I mean, the basic process is from the blood collection, we're able to separate and concentrate out that exosome and nanoparticle fraction of the blood. And the final product is then tested for microbiology contamination and endotoxin for safety. And the final vials are frozen and shipped back to the doctor for use. And on the product development and quality control side, we check products for nanoparticle concentration and protein concentration on a fairly regular basis. But with each collection, where we are required to test for microbiology and endotoxin concentration.
Yeah, and there's another question that's related. Is there something that we send to your lab? At least the way we do it. Again, I don't want to, at least for now, we may be the only one doing it. We send you a kit, a special kit that's easy to use, where you draw the blood into a cylinder. It's labeled easily. Your staff that can do phlebotomy do this. Anybody that does phlebotomy can do this. And then we have a special designed FedEx box where you just push a button, throw the blood in there with the labels, and it cools it down for 3 days, and it's picked up. We arrange for it to be picked up. It's shipped to our lab. So it's really a easy process. You don't have to spin anything in your office like PRP. Simply have someone draw the blood, package it up, and ship it to us.
So we had a few patients where we had a mobile phlebotomist go to the home after the kit was delivered to the phlebotomy people, and then they drew the blood and then saw their orthopedic surgeon 2 weeks later to get the injections.
Yeah, it's about 10 days or so. You get the therapeutic back.
Okay, can you administer exosomes through epidural or intrathecal?
I'm gonna let you handle that, George.
Sure. So I know there's a lot of companies that do that. Fountain Life, who's the sole provider of the Organicell product, with PM&R specialists, they could do that, but we don't do any epidural injections or intrathecal injections. And the reason is because these exosomes can cross the blood-brain barrier. We're going to be presenting at a conference in 2 weeks in Vegas, and we're going to be discussing focused ultrasound of the brain to mobilize exosomes that you inject IV to specific areas of the brain, like the amygdala, the abducens, the basal ganglia, to draw these exosomes to those areas to have it work in its anti-inflammatory effects. So you don't really need to do something as invasive as intrathecal or epidural. I never used to like that. Even worked in the ERs when I used to work in the ERs. Sometimes you get punctures of your CSF fluid, you need epidural patch. It's just, you want to make life easy for you. I mean, I know there's some, interventionalists who do this, but you don't necessarily need to do that. You can just do an IV and it crosses your blood-brain barrier.
George, here's another question for you, because you've treated so many patients with exosomes and now autologous. Other than cancer, are there any other contraindications, Dr. Bano's asking, that you would think that you want to avoid?
Pregnancy. I never like to do anything in pregnant women. Active cancer, active infections. I just don't, well, actually, no, because COVID was a really bad infection and we were injecting in COVID. I think we have a list of the contraindication, but this is your own blood. So this is not someone else's. So you're giving your own blood back. So, you know, I would just say active cancer right now and pregnancy.
Okay, very helpful.
There's a
Let's
question
see.
from Ray Zoller. Ray, glad you could join us tonight. Does Organicell own any protected IP around its processing of the exosomes?
Sorry, what was that question?
Does Organicell own any IP?
Yeah, I'll take that. Yes, we do. Yes, of course. We do have patents pending and filed for patents and own patents, and we do have IP on this. Yes, I mean a lot of work has been done to be able to do this, so that we do have intellectual property and know-how beyond the patents. We've learned a lot, and I want to congratulate Michael and Dr. Mike Bellio, Dr. Bellio and Dr. Golub. I mean, there's a lot of. I mean, it sounds easy isolating exosomes. It's not for something you do, you know, in the office or on your kitchen table. So we have patents and we have a lot of know-how that aren't even in the patents about how to do this.
So another good question, is there an autoimmune disease contraindication to using exosome therapy?
Autoimmune, I mean.
So we have a lot of patients who come in with rheumatoid arthritis or diabetes or Crohn's disease or ulcerative colitis. And a lot of these autoimmune diseases are sometimes due to an overproduction of your own immune system, interleukin 6, TNF alpha. We've shown that exosomes reduce the inflammatory response, and by creating the monocyte from M1 to M2. They're excellent for autoimmune patients. Stem cells have already been shown to almost cure Crohn's disease and colitis in some scientific studies. And think about the secretion of the exosome. The secretion of the stem cell is the exosome. So absolutely, they're great for autoimmune diseases. Something called therapeutic plasma exchange, in addition, is also excellent for autoimmune disease.
Yeah, I want to cover the question about inflammatory lung disease. There's a lot of interest and emerging evidence that exosomes can really be powerful in inflammatory lung disease. The prototypical one, there's a doctor at the University of Chicago, I think it's Marilyn Glassman, who is the chair of medicine, and she spent her entire life studying and trying to come up with treatments for idiopathic pulmonary fibrosis, which some of you may know is a relatively rare but universally fatal disease where you get at middle age and you die within 3 to 5 years from progressive fibrosis of the lung. And she's got preclinical studies in animals that shows that exosomes actually stop the pulmonary fibrosis and maybe reverse this, and has a small number of humans that have been treated. So there's a growing interest and belief that in inflammatory lung disease, whether it be viral like COVID, where we have data, as George said, or whether it be IPF, which is a very bad inflammatory disease, exosomes may be the magic bullet. It's early days, but it's very exciting.
So we have a bunch more questions. Where can we access trials? You just go to PubMed and you can just put in Organicell and you can see the clinical trials.
Mm-hmm.
How do you get started using exosomes? You can send us an email. You can go to our website, Organicell.com. We have a whole team that can help turnkey your office into starting using exosomes.
Do we have a contact based on this webinar,
Jill?
I
just added it, George and Harry. Hi everyone, I'm Jill Swartz.
Yeah, hi, Jill.
To the chat where you can actually, Tammy Banna, who's our clinic product specialist, is on this webinar, and I also put a link to her Calendly and to her direct number, and she can set up a call with any of the physician presenters here. She can answer directly lots of calls. She's training doctors all across the country on autologous exosomes, and that's the best first starting point to get reconnected with us to learn more.
So how many treatments do you think you're going to require for these autologous exosomes?
Yeah, George, I'm going to again.
So from our experience, you need about at least 2 treatments, sometimes 3. Right now, we're starting to get into phase 2 trials to find out how many billions of exosomes do you need and how many months apart you need to give it. Right now, we're doing it, once a season. We have several protocols that we put together in our trials, and that's what we're following.
It obviously depends on the clinical
Yeah,
indication.
yeah.
And as we do studies, as George says, we're going to work on actually optimizing the dosing schedule.
Yeah. So for example, hair might be different than something else. So it does depend on the clinical condition, certainly.
Right. I know Dr. Ziering is on the call, and he has a very large practice in New York, Connecticut, and in California. And he's been working with us for a while using aesthetics. So not for inflammation, just using it for aesthetics, hair. It works great on hair. It works great on your skin. So there's many different uses, whether you use it IV or inhalation for some diseases. That was a question. All of our trials that we've done, IV seems to work great, but the inhalation we're just starting to use now. And we believe that already from research done at University of Chicago with Dr. Glassberg using exosomes for idiopathic pulmonary fibrosis in some early studies, we believe that the inhalation can take your lung tissue and turn it from a fibrotic lung into a normal lung again. And that's some of the research we're going to be doing hopefully soon with the Mayo Clinic. We were approved for a trial with the Mayo Clinic, and more research coming out of University of Chicago. So there's a lot of research happening with exosomes, but the main thing from tonight is, autologous exosomes are stronger than regular PRP, and you're able to use them without any worry.
Do we cover all of them? We may have.
I think we have.
Oh, the one question I'm going to cover is any thoughts on allogenic, because we kind of touched on it and we alluded to it. And George covered one point. So in 2019, the FDA came out with a statement. This isn't a law. It's not a criminal law. It's not this isn't like, something Congress passed. This is a regulation saying that allogenic exosomes are a biologic therapy that require FDA regulation. And what that means is the FDA is saying that they believe that they should get companies or should pharma companies like us or anyone should get FDA approval before marketing. We don't really market allogenic exosomes for specific uses. We make them, but we never say to doctors what to do with them. The attention that's somewhat constructive is physicians with their license can use anything they want to treat patients under the quote practice of medicine. So there's been this time-honored tension between regulations and what doctors can do treating patients. So it's a gray zone between the regulations and doctors' rights under the practice of medicine. What George is saying so eloquently is one of the great things about the allogenic exosomes is there's no gray here. There's no tension. It's clear
autologous, you mean,
because they're autologous. Because of their autologous, you simply don't have to worry about this uncertainty, and they work as well.
We can provide any of the physicians on the call with some information, so they'll be able to get these and use them on their patients. There's a question here: any recommendations for a GI provider that uses exosomes? This is new. A lot of doctors don't know about the autologous exosomes. So we'll be, if you give us a doctor, we can contact them, explain to them about the product, and they can use it. And if you have colitis, I would absolutely try this because it's already worked on many patients.
Yeah, I'm very excited about exosomes and inflammatory bowel disease. I think it's a great thing to, that we're all interested in. Maybe
there's a question about ALS. I just want to address that.
We've
had several people who we're treating with ALS, but we're finding with ALS that it seems that it may be due to a retrovirus, that's indolent virus in you, and if you think about what do the exosomes do to reduce inflammation from viruses, they could help, but again, this will have to be an N of 1 clinical trial. We have to be very careful. We can't just start treating diseases. It's just not healthy for a company to start making all these claims. That's how you get into trouble. So, we want to stick to what we know, and we want to stick to what's been working. So right now we're using for inflammation, arthritis, and all those indications that Dr. Leider presented before. But autologous exosome is just like PRP. So whatever you're used to using PRP with, you can now use the second generation PRP product.
I think we've covered just about everything. And we've had up to like 80 people on the call tonight. Incredible for an evening for everybody to be on the call. I think it demonstrates the interest in exosomes, in regenerative medicine, specifically autologous exosomes. And I want to thank the panel. I get the pleasure of working with these guys every day, but they're doing the hard work to help bring this science to you. And for any of you that will be next week at the A4M conference too, as we said, there's two great organizations. But if you're going to be out in Las Vegas, we'll be there. We'll have a booth and be happy to talk to you there too.
And we'll also be sure to do a follow-up email to everyone who registered and make sure you get a recording of the webinar tonight, and we'll follow up with details and the presentations.
And
Yeah,
my
one
final
last
comment.
thing I wanted to mention is we
Yeah,
have a few of our medical advisors who are listening to the call. One, Dr. Carolyn DeLucia, who is an expert in female intimate health, and she's been using exosomes for a while. So we have a lot of doctors in dermatology, orthopedics, neurology, who work on our advisory board. So if you have specific questions regarding like the ALS, the MS, the Parkinson's, we can definitely have someone reach out to you. We have a top neurologist in the country at UCLA doing a lot of experiments with and research on exosomes right now. So we're really a science company. And we like to really provide the science. Dr. Golub joined us. He's been with the NIH for years with a lot of expertise. Dr. Michael Bellio is really incredibly amazing. This has been his whole PhD. And for those of you who are PhDs, it takes a lot of work to get a PhD. And this is his whole life research is biology and stem cells and cell therapy. And then Dr. Leider, thank you for running this.
Oh, my pleasure. I mean, working with you guys, great.
Dr. Leider, sorry.
That's right, Leider. But the last
You're a true leader, so.
Leider, but the last person I want to thank is Jill, our Chief Product Officer. She organized this, put dozens of hours in working with everybody, and made this possible tonight. So thank you, Jill.
I had one more question on here. Jill, can you ship this outside the US to like Canada or Puerto Rico or Mexico?
And the question, the answer is yes, we can. I mean, for Puerto Rico, especially, is a U.S. Region, and yes, we've shipped PRP. We've shipped it all over. So yes, the short answer is yes.
If somebody wants to get exosomes tomorrow, is there a place they can call, or just go right to the website?
Yeah, I actually put Tammy's number in the chat, and we could set up a calendar. And absolutely, you can go to the website. There's a number on the website as well. But
what's
Tammy
the website?
And myself,
what is the website, Jill?
It's www.organicell.com.
Okay, thanks.
Just want to echo quickly what Tammy answered to the international. It depends on the particular countries, and some countries we haven't researched yet, but we can find out for you.
Okay, I think we should wrap it up. So thank you. I guess if you remember one thing, that autologous exosomes are very potent, anti-inflammation, pro-cellular regrowth and repair, and autologous exosomes are a way that you can really think about applying this to your practice with confidence, both in their potency, but regardless of age, but also it's a safe harbor, if you will, from an FDA standpoint.
And I'll add one thing to that, and it's just because I've been in this space of regenerative medicine for quite some time, really deeply seated in the PRP world. This whole space is growing so quickly. So this isn't a matter of information that you may or may do in your practice. It's information you need to know, because this is the next frontier of medicine. This is the way regenerative medicine is going. PRP was yesterday, as again, it's a tool that is not going to go away, but autologous exosomes and exosomes as a whole is the next frontier. So we need to be armed with this information, because these consumers are driving the demand and they're going to be asking for this. So it's you need to be armed with it. So we're so grateful that you spent a full hour out of your evening
Yeah,
to.
I just want to make a
listen.
comment. So I've been working with Jill Schwartz for almost 15 years with PRP when PRP was big. And I never thought she'd switch from PRP to a different product, but she did, because it's second generation PRP.
Yeah.
Yeah.
All right. Well, thank you guys. And again, you can talk to Tammy's in the chat. That might be the easiest way to contact us. Our website. George is a wealth of information on the real world. So yeah, we're here to answer your question. Just thank you for being here. It's an evening. It's time out of your life, and really appreciate it.
Thank you.
Okay,
Bye
take care.
bye.
Good night.